Okay. Welcome to the Spring 2021 Core Facility Seminar Series. Today we will be featuring the Human Magnetic Resonance Center, or hMRC. I am Lisa Korpieski, the Director of Communications and Marketing here at the Institute for Applied Life Sciences. In today's seminar, we will hear from Rajakumar Nagarajan, the MR physicist in the hMRC. He will tell us about the state-of-the-art imaging technologies available through the only research-dedicated 3T MRI system in Western Massachusetts. In addition, we are excited to have Gwenna Layak, Assistant Professor in Kinesiology and IALS, as our guest speaker. We hope that with these bi-weekly seminars, you will discover what great resources that the centralized UMass Amherst core facilities offer to our campus community, the New England region, and beyond. Next, just a few housekeeping items. The seminar is being recorded, as you just heard. If you miss any part of the seminar or would like to forward it to someone who could not attend, there will be a replay of this and all previous seminars in this series on our website. We will put the link in the chat. I recommend you set your view mode to speaker. Please stay muted during the talks. We will save the Q&A until the end of the presentations. During that time, I welcome you to unmute yourself and ask your question. So now I would like to introduce you to Andrew Vernard, Director of our centralized UMass Amherst core facilities. Andrew?
Thank you, Lisa. Good afternoon, everyone, and thank you for joining us today for the 8th and second to last seminar of the Spring 2021 Core Seminar Series. We hope you've enjoyed our programming thus far. We welcome your feedback and any suggestions you might have for future content and seminars. As Lisa mentioned, my name is Andrew Vernard, and I'm the Director of our centralized cores here at UMass Amherst. Our state-of-the-art labs, instrumentation, equipment, and most importantly, world-class experts are helping researchers drive discovery and pursue scientific endeavors across all disciplines. From sending samples for analysis or designs to be 3D printed, to becoming a trained user in advanced spectroscopy techniques, there are ample opportunities for engagement with us. The core facilities are open to anyone from students through senior scientists, regardless of your affiliation, including researchers from other academic institutions and commercial operations. To help access our cores, there are several funding opportunities. For those IALS investigators on the call, core credits and center seed funding is available. For our external users, the Mass Innovation Voucher Program subsidizes usage for small companies based in Massachusetts at up to 75%. To date, the program has awarded more than 600 vouchers, which represents about $5 million in total project costs for our cores across all 5 UMass campuses. Our goal is to be your partner by expanding your research capabilities and productivity. Please feel free to reach out to me or any of the core facilities directly if you have questions or would like more information about our capabilities and how we can work together. It's now my pleasure to introduce Dr. Rajakumar Nagarajan. Raja joined UMass and the hMRC from the University of California, Los Angeles just about 5 years ago as one of the first staff members in the facility. He's been instrumental in providing support for the MR spectroscopy community, including training of many students, both directly through the facility and via an MRS course that he's taught a few times over multiple semesters. Raja has also represented the hMRC and UMass on the international stage by presenting MRS work generated in the facility at the annual ISMRM conference. We are grateful for all of Raja's contributions to UMass. Raja, along with Elena Bliss, the hMRC MR technologist, Dr. Jackie Kurland, our longest tenured interim director, and Dr. Gottfried Schlag, the hMRC scientific director, are responsible for the only human research magnet in Western Massachusetts, a resource with tremendous potential that I'm glad you'll be hearing a bit more about today. With that, I'll turn it over. I'll thank you for joining us again and be available during the Q&A if you have any questions. Thanks, Raja.
Thank you, Andrew. Welcome, everyone, and thank you for joining us for today's seminar. My name is Rajakumar Nagarajan. I am the MRI physicist at the Human Magnetic Resonance Center. We call us hMRC. Let me give you a little bit background about the hMRC. hMRC provides state-of-the-art, non-invasive whole body imaging and spectroscopy technologies for academic and industry-based research. This is the only dedicated 3D MRI/MRS system in Western Massachusetts, and it has multinuclear capabilities. That means in addition to proton, you can do phosphorus and carbon. MRI produces 3-dimensional detailed anatomical images, where MRS provides biochemical information. We have Siemens 3T Magnetum Skyra MR scanner, which is 70 centimeter wide bore. When you see in the hospital, you can see 60 centimeter bore, which sometimes patients, they feel claustrophobic. This 70 centimeter wide bore, which will minimize the claustrophobic. And the kinesiology department, they are working mostly on the exercise protocol. So 70 centimeter is really useful for their research. Our scanner will produce highly detailed anatomical imaging of any tissue with a spatial resolution of 1 millimeter. And also the scanner is capable of doing spectroscopy to measure the biochemistry and the metabolism with a concentration of 1 millimolar. And also the scanner is capable of doing functional MRI, which is called bold blood oxygenation level dependent. So we have strong users from psychology, communication disorder, and the Bay State Hospital. They are doing functional MRI, which will measure and mapping the brain activity in the real time as well as safe. And also you can do DTA imaging, which is called a diffusion tensor imaging, which will find out the location and also estimate the fiber track in the white matter. We have ancillary equipment for data collection like ergometers, bowl screen, eye tracker, audio and physio recording, EEG, transcranial stimulation, numerous response and feedback devices. hMRC offers the following services: implementation and validation of novel MR spectroscopy sequences. It's a single one-dimensional, multidimensional spectroscopy sequences. And we are providing technical assistance on designing the experiment, data processing, and troubleshoot. We are assisting users on NIH and other grant submission. We are providing seminars for users and graduate students. hMRC offers Introduction to MRI and Spectroscopy courses in collaboration with the Kinesiology. This is a 3-credit course offered every spring. We have collaboration with the Bay State Hospital clinicians on the MRI and the MRS project. We are providing safety education and training to users. Our MR technologist, Elena Bliss, she is giving training. And we are assisting graduate and undergrad students to present their work in the meeting and conferences. I just wanted to show you a few images from our facility. So the figure A, which is the anatomical information of the brain, which is a 3D image with a spatial resolution of 1 millimeter. So we have a very beautiful head coil with the 20 channel, 32 and 64 channel, which you can see anatomical as well as the pathological information. And the figure B is functional MRI, where you can see the mapping of brain activity. For example, if you tap a finger, where you can see the activity in the cortex, which you can see it in the real time. The third figure is the liver MRI. See, for example, if you want to calculate fat percentage for non-alcoholic fatty liver patients, so that is a 15 seconds breath hold sequence. So with a very nice spatial resolution. Figure D is the breast MRI, which you can see how the breast density can be calculated. And the last figure is a muscle MRI, which provides muscle function and structure. In addition, we are doing elastography. And this slide shows few examples from MR spectroscopy. Figure A is a muscle MR spectroscopy, where you can see on the time series real time during the rest, exercise, recovery, how the phosphorus metabolites are changing during the real time, like a PCR and inorganic phosphate. And a figure B, you can see the small box, which is the location of the wax cell from liver. You can see how the lipid and the choline is changing for any non-alcoholic liver fatty patient, where you can see how these biochemicals are changing. Figure C is a brain tumor wild type, where you can see elevation of choline and creatine and decreased N-acetyl aspartate, 2-hydroxyglutarate. And figure D is the spinal spectroscopy. For example, if you want to do spectroscopy in the spinal spondylosis patient, where you can see and different metabolites, changes you can see for spinal spondylosis, you can see elevation of choline. And this figure E is a invasive ductal carcinoma, where you can see elevation of lipids and choline. Like that, you can do spectroscopy in the whole body. And hMRC housing the following coils. We have 20 channels, 32 and 64 channel head coils. So higher the channel is, better signal to noise ratio. And we have few surface coil. So this one is a phosphorus, and this one is a proton, phosphorus, and the carbon surface coil. So kinesiology department is mostly doing exercise protocol to measure deoxy, myoglobin, phosphorus, and glycogen. And we have multinuclear carbon and phosphorus coil where you can map phosphocreatine, ATP, glycogen in soleus, tibialis anterior, and gastrocnemius. And this is the flux coil. For example, if you want to see liver MRI, prostate MRI, you can use this coil. And this is a breast 8-channel coil, and also the knee coil, which is in case if you want to do any cartilage imaging. And our hMRC leadership, Andrew Winard is our director, core facilities. Jackie Corland is our interim director. Godfrey Schluck is our scientific director. In addition to me, Elena Bliss, she's our MR technologist. With that, it is my great pleasure to introduce the guest speaker, Dr. Gwenna Layak. He's an assistant professor in the Department of Kinesiology at UMass Amherst. His research focuses on the development of novel in vivo imaging techniques that use MRI and MRS for the study of muscle. He explores pharmacologic, dietary, and exercise-based strategies to mitigate the deleterious consequences of aging and various diseases. Example, COPD, PAD, and type 2 diabetes on muscle function with the ultimate goal of improving mobility and reduce disease burden. He used a translational approach from the cellular level to the whole organism in humans, which encompasses cutting-edge MRI and MRS coupled to ultrasound Doppler, near-infrared spectroscopy, high-resolution respirometry in muscle fibers, and a host of molecular and cellular biology techniques. He has published more than 70 peer-reviewed manuscripts and secured a few NIH grants, and also presented several conference abstracts. With that, I will hand over to Dr. Layak.
Well, thank you, Raja, for this introduction. Let me share my screen and I can get this going. Here you go. And I think now you should have access to the slideshow. Let me just make sure that. All right. Again, thanks for the introduction. And so today my talk is going to focus on a couple of applications which I think are relevant to the expertise and the equipment that is available in the core. But before I start my talk, I really want to thank Raja and Elena for contributing to the work that we do in the lab. And thanks to their expertise, we've been able to collect high quality data in the last 2 years. So again, thank you very much for your help with our work. So I didn't know what would be the audience for this talk. So I decided to go with a broad introduction. So the skeletal muscle account for about half of our body weight. And so you have about 600 skeletal muscles in the body, and they are involved in multiple functions, such as locomotion, breathing, posture. And most of the time, you actually don't think about this skeletal muscle unless you go for a run and the next day you feel very sore. Or if you have some myopathy, so a disease of the muscle. So for instance, Duchenne muscular dystrophy is one of the most studied myopathy. And it's kind of a devastating disease, which affects a specific gene that codes for this protein called dystrophin. And that leads to abnormalities of the skeletal muscle, the respiratory and the cardiac muscles. As I was mentioning, it's really a devastating disease because these children usually don't reach adulthood. But skeletal muscle dysfunction is also important in many chronic conditions, such as PAD, type 2 diabetes, not part of the primary etiology of the disease, but it's going to contribute to the prognosis of the disease and also to the poor quality of life of these patients. So it is an important topic to study. So let's now move on to the techniques related to magnetic resonance. And the way I like to look at MRI is that it's kind of eating this French pastry that's called a millefeuille or 1,000 layers. And the reason I'm taking this example is because people are going to eat this pastry in many different ways. Sometimes they start with the sugar on the top, the custard in the middle, or all together. It doesn't matter. It's always tasty and excellent. And I think MR techniques are kind of the same. Like you have many different ways of using a scanner, and it's always going to provide you with some very insightful information. But we can summarize these techniques into two main categories, spectroscopic techniques and imaging techniques. And for the first part of my talk, I'm going to focus on MR spectroscopy. So the way I used to present that to our participants in the lab is that, an MR scanner is really just a transistor radio, just a little bit more expensive. You need an antenna. In the MR field, we call that a coil. And so in the hMRC, you have these different coils. So this one is a surface coil to look at phosphorus. This bird cage is actually for carbon-13 and phosphorus. And then this flex coil can be used for many different configurations, but also for proton spectroscopy. So you've got the transistor radio, the antenna. Now you can pick your radio. But with MR, you have some restriction. So you need to choose a specific frequency that is going to correspond to the Larmor frequency, which I have here. And this frequency is proportional to the magnetic field, so this B0 here, and the gyromagnetic ratio, which is specific to the nucleus. And so I have here the values for proton, carbon-13, and phosphorus at 2.9 Tesla, which is the scanner that we have here in the hMRC. So once you have all of that, this is the type of spectra that you will be able to collect in the skeletal muscle. So you can see that when you use proton spectroscopy, you have quite a large number of metabolites to look at. With carbon-13, you're usually going to focus on glycogen. And then with phosphorus, you're going to have all the phosphate-containing metabolites in the skeletal muscle. So let's look at this phosphate first, because that's really the nucleus that has been the most studied in the skeletal muscle. And so I have here a couple of nuclei of interest. And one thing that I want to mention is that you can only get a resonance with an MR scanners on a nuclei that has a non-integer net spin. So you see that for the phosphorus, it is half. Then there are two parameters that are important to know. The abundance, you see that phosphorus is well abundant in the environment and in the body, and the sensitivity. So the sensitivity is usually expressed in relation to a proton. So proton or water is going to be at 1. And you see that the phosphorus is only 6%. But again, because it's fairly abundant, this is actually going to be a nucleus with a high signal. And I have here the picture again of the coils that are available at the core facility. So where should we start to study muscle function? Well, we're going to take a trip down to the cellular level, and we're going to look at the myosin head here, which is kind of the molecular motor of the muscle contraction. So this myosin head contains an enzyme called the myosin ATPase. And what this enzyme does is that it breaks down ATP into adenosine diphosphate and inorganic phosphate, so this ADP and PI. And the energy that is generated by this reaction is going to lead to mechanical work, so the movement of this myosin head. But you only have a limited amount of ATP in your body, so probably just for a few seconds. So you need to replenish this ATP store, and for that, you're going to use phosphocreatine. And that's mediated through this reaction with the creatine kinase. So you use phosphocreatine and ADP to regenerate ATP, and you produce free creatine. But similar to ATP, this phosphocreatine is in limited amount in the skeletal muscle. So you're going to use another process to regenerate or resynthesize ATP, and that's the oxidative phosphorylation, which occur in the mitochondria. It's really a complex process, but it needs two things to be able to run and produce ATP. One is oxygen. And so you need oxygen that comes from the circulation. So it's released by the red blood cells, cross the capillary membrane and gets into the myocyte. There, the myoglobin binds to the oxygen and is going to bring this molecule of oxygen to the mitochondria. The other substrate that oxidative phosphorylation is going to need is called the reducing equivalents, NADH and FADH. And those are produced through the breakdown of glycogen into glucose, and then this glucose enters the glycolysis. It's broken down into multiple intermediates, which we're not going to go over today. But one of the byproducts of this glycolysis is actually lactate and hydrogen ions. And you also have a small amount of ATP during glycolysis. And what is very interesting when you think about MR techniques is that all the reactions that I was mentioning over here can actually be measured by phosphorus spectroscopy. You can also quantify glycogen levels with carbon-13 spectroscopy, and then you can quantify lactate and myoglobin oxygenation using MR spectroscopy of proton. You can also quantify using some imaging technique, tissue perfusion, and hemoglobin saturation. You can even quantify muscle volume and the muscle characteristics, again, using some MRI techniques. So you see all these techniques are non-invasive, and you can obtain all this information in this schematic just using MR techniques. So let's now look at the technical aspect of measuring phosphorus metabolites in the skeletal muscle. And so this is the typical spectra that you would obtain in the skeletal muscle with this technique. And there are a couple of things that I want to highlight in this spectra. So one is the dispersion of the metabolites. So look at this scale here, that's in ppm. You see that it goes from minus 20 ppm to about 5 ppm. During a high intensity exercise, you would see an increase, sort of peak over here corresponding to the phosphomonoester. So you have a fairly large spread in the metabolites that you can observe with phosphorus and MR. And you already have only a handful of metabolites. So you have 3 ATP, phosphocreatine, phosphodiester, inorganic phosphate. So you have roughly 6 metabolites to look at over a fairly large range. And that makes the quantification of these metabolites fairly easy in comparison to proton spectroscopy, for instance. And so the first step you would do in an experiment would be to quantify the area under the curve for these different metabolites, because it's going to be proportional to the concentration of this metabolite. Another interesting parameter in this spectra would be actually the chemical displacement of the inorganic phosphate. What this means is that when the muscle acidifies, the inorganic phosphate is actually getting closer to the phosphocreatine. And what biochemists measured very early on in the development of this technique was that this chemical shift, which I have illustrated here, was actually related to the pH of the solution. So they established this titration curve, which we now use to accurately estimate the pH in the skeletal muscle, or the intracellular pH. In addition to this, you can also quantify a couple of compounds based on known biochemical reactions in the body and using some dissociation constants. So you will be able to quantify the concentration of adenosine diphosphate, inorganic phosphate deprotonated, adenosine monophosphate, and the free Gibbs energy of ATP. So now you're starting to have a fair amount of information, but that wouldn't be that useful. It turns out that at rest, you really don't have much differences between people. Whether you're Lance Armstrong or couch potatoes, this spectra is roughly going to look the same. What you need to do is to induce a stress on this muscle. You need to have an exercise that is going to induce a metabolic disturbance. And now you can look at the metabolic function and the skeletal muscle function. And so researchers have developed many different types of ergometers to actually induce this stress in the MR scanner. So I have here a couple of illustrations of the ergometers that have been published in the literature. And that might seem trivial to actually build an ergometer in many kinesiology labs. It's fairly easy to build to build an ergometer. But this is actually more challenging when you are in this large, in a narrow scanner with a very large field strength. So you really limit the number of materials that you can use. And just to give you an idea, so this ergometer here, which is commercialized by Lodi, costs about $50,000 to $60,000. So quite a significant investment. Here in the hMRC, Dr. Kent collaborated with some faculty member of the bioengineering department, and they built this knee extension ergometer to look at the quadriceps muscle. In our lab, we use this plantar flexion system to look at the calf muscle, and it is MR compatible as well. So we can have people performing a plantar flexion exercise in the scanner, and meanwhile, we quantify the power output with this ergometer. So once you have an ergometer, you can induce a stress on the muscle, and you can see the metabolites now changing. So here, this large peak, that's the phosphocreatine. And you see that as soon as you start contracting the muscle, the phosphocreatine goes down. And then as soon as you stop, the phosphocreatine is going to recover quickly. If you look at this peak on the left, that would be the inorganic phosphate. And as soon as you start to exercise, you accumulate inorganic phosphate in the muscle. And depending on the intensity, you can even see a splitting of this peak. And that usually means that you're working at a fairly high intensity. What doesn't change is actually the ATP concentration. For most exercises, that stays fairly constant. So when you perform this exercise, you're also going to be able to quantify the ATP resynthesis from three main pathways, three key pathways of the skeletal muscle. The creatine kinase, so the resynthesis of ATP through the creatine kinase reaction, glycolysis, and oxidative ATP synthesis. And one parameter that has received a lot of attention is this mitochondrial phosphorylation capacity. Sometimes it's called muscle oxidative capacity. Basically, it's how much your mitochondria can generate in terms of ATP. What's the capacity of the mitochondria to produce ATP? If you have an ergometer that can quantify the mechanical work, you can then also quantify the ATP cost of contraction, which is actually also a very important parameter. So if I were to compare the skeletal muscle with a car, this mitochondrial capacity will be the size of the engine, how much horsepower you've got in the engine, and the ATP cost of contraction would be your gas mileage. So both of them are important if you want to know if your car is actually a good one. And so now I'm going to illustrate some of the applications that I've seen in the literature with this technique. And I'm going to present data in the context of aging, because that has received quite a lot of attention. So let's start first with the mitochondrial capacity. So here what you have displayed is the phosphocreatine concentration after an exercise. And so here this x-axis is the time. So as soon as you stop to exercise, the phosphocreatine is recovering resynthesize the phosphocreatine, and you can fit this recovery with a mono-exponential function, and you obtain this parameter called the time constant. And this time constant is inversely proportional to the mitochondrial capacity. In other words, if you have a very short time constant, you have a very high mitochondrial capacity. So if you recover the phosphocreatine quickly, that means that you have a high mitochondrial capacity. And so using this approach, Dr. Kent, who is in the Department of Kinesiology, so she has performed many studies on this topic, but she published this review looking at the recovery of phosphocreatine in different skeletal muscles. So the knee extensor, plantar flexor muscles, upper extremity, and dorsiflexors. And what you can see from this review is that there is really an heterogeneous effect of aging on the mitochondrial capacity. Just look at the dorsiflexors. Turns out that as we get older, the dorsiflexors get better in terms of mitochondrial capacity. If you look at the knee extensor, that's the opposite. So there is clearly, different response to aging in the locomotor muscles and also in the upper extremity muscles. And that seems to be dependent on the muscle activity. But don't think that because you get older, that mitochondrial capacity is going down. This is what those data are showing. Now to illustrate the importance of the ATP cost of contraction, I'm actually going to use some of my own data. So in this study, we looked at the plantar flexor muscle. So we had people doing a dynamic plantar flexion exercise. And we had 3 groups of participants, a young adult group, an older adult group, and the last one, which we call the oldest old. And this name might sound a little odd to you. Turns out it's the term that is used in the field to characterize people that are between the age of 80 and 85. It seems that's about the age that most people can reach if they don't develop some sort of medical conditions. And so if they stay healthy, they can expect to live about 80 to 85 years. If you go above 85, that means that you probably have some genetic predisposition in your family. So we were lucky enough to be able to recruit 10 participants with an average age of 84. And so this figure here represents the ATP cost of contraction in these 3 groups. And what we found was that as you get older, the ATP cost of contraction was getting higher. And in fact, it was the highest in the oldest old, actually 3 times higher than in the young adult. In another study, we found that this was mainly driven by a greater energy demand from the ionic pump between the contraction. All right, so I have presented, I think, a lot of the advantages of this technique, but there are a few pitfalls. So compare to biopsies, NMR techniques, at least Phosphorus, tends to have a lower sensitivity. You cannot detect metabolites, or it's hardly detectable if the metabolites is lower than 1 millimole. And one consequence of that is that you're not going to study single fiber or fiber bundles in the scanner. You also need a stimulus, either an ischemia or an exercise. Another issue is related to the quantification. There are different theories of the metabolic control for the mitochondria. And so different labs are using, again, there are only a handful of theories, but different labs may use different assumptions. And this might lead to slightly different rates in their estimation of ATP resynthesis. Now, where do we see this technique going? Clearly, in the last 10 years, there's been a lot of studies on spectroscopic imaging. And I was trying to illustrate that here, where you can see actually a map of the phosphocreatine concentration in the lower leg. But as you can see from the scale on the right, there is still some work to do. in this with this technique is phosphocreatine levels 60 millimole in the lower limb. That's just not physiological. So there are still some improvement to make there. Another technique that we're actually trying to develop in the lab in collaboration with hMRC is this non-invasive biomarker of mitochondrial content. So it turns out that there is a small peak next to the inorganic phosphate peak that seems to be related to the mitochondrial content. So it's still very early, but we're trying to validate these little metabolites as a biomarker of mitochondrial content. We'll see if this holds true. Let's now focus on proton spectroscopy, and you see here that it is abundant, very high sensitivity, so that makes it a very nice target to perform spectroscopy on it. And here we're actually going to focus on the left-hand side of my conceptual framework. With this technique, with proton spectroscopy, you can quantify lactate and also myoglobin oxygenation. And actually for this talk, I'm really just going to focus on myoglobin oxygenation. And we kind of see it, we measure the oxygenation level in the skeletal muscle in a kind of counterintuitive way. We look at the deoxymyoglobin, not the oxygenation, the deoxygenation of the myoglobin. And so what you see here is that at rest, we actually don't see a signal. The myoglobin is almost fully saturated at rest, and so it is below the threshold for detection. But as soon as you induce an ischemia, you can see this peak rising. And within 5 to 6 minutes, you start seeing a plateau. And so this very large peak here, that's the deoxymyoglobin signal. Depending on the parameter of your sequence, you will be able to see a second peak, the deoxyhemoglobin. But again, this is really dependent on the parameters of the sequence. Most of the time, we do not see it. If you look at the scale at the bottom, look at the ppm here. We're actually 74 to 78 ppm away from water, so very far from water. And this is a chance because the concentration of myoglobin is very low in the skeletal muscle, only a few millimoles. And because we are so far from this large signal, which is the water signal, we still have a chance to see this deoxymyoglobin signal. So what we've been able to do here is actually to see this deoxymyoglobin signal during exercise. So this is actually a data set that has been collected here at the hMRC in a COVID-19 survivors, which is a project that members of my lab are leading here. And so you can see that the deoxymyoglobin peak is going up as soon as they start to exercise. And we had a brief period of ischemia after 3 or 4 minutes of exercise. So you see the peak going up over here. That's the ischemia. As soon as you stop the ischemia, so you release the occlusion, you see that the deoxymyoglobin peak is going down. So you're reoxygenating the muscle. And so to illustrate the usefulness of this approach, I'm actually using data that I collected in collaboration with Tony Donato and Tracy Freck when I was at the University of Utah. And with these collaborators, we recruited patients that suffer of systemic sclerosis. And if you've never heard of this disease, that's an autoimmune disease. That's actually a rare disease that is characterized by a progressive destruction of the capillaries. And so these patients tend to show up with Raynaud's syndromes, digital ulcers, and some skin thickening. And you might think that this is actually a fairly benign disease, but it turns out that when someone is diagnosed with systemic sclerosis, the life expectancy is usually only 5 to 10 years. So it is actually a devastating disease. And the treatment are actually, currently there are no real treatment for that. These patients are provided vasodilators to improve the circulation, but there is currently there is no cure for this disease. That tends to affect women predominantly, and so that's reflected in our recruitment over here. So we had these patients performing a plantar flexion exercise in the scanner. The first time we measure muscle metabolism using phosphorus NMR, and specifically we looked at the PCR recovery, which you have displayed over here. And then we had them coming back and performing the same exercise while we measured myoglobin oxygenation. And so looking at the phosphocreatine recovery, so which is displayed over here. So you have the control group here, and then the systemic sclerosis patients there. You see that the recovery was much slower in these patients. That's a very slow recovery. And just from this, you would think that, well, they seem to be having also a mitochondrial dysfunction. But because we had collected this data on the myoglobin oxygenation, we also saw that, even at rest, they had some level of deoxygenation. The myoglobin wasn't fully oxygenated and went way down during the exercise. So there was a significant difference in myoglobin oxygenations between the control and the systemic sclerosis patients. And this suggested that the oxygen supply was likely limiting the PCR recovery kinetics. So the capillary destruction, which is typical in this disease, was leading to this limitation in oxygen supply, eventually limiting the production of ATP by the mitochondria, which also explain the poor functional capacity of these patients. So I think this is a very good illustration of the complementarity of these two techniques. If we had used only phosphorus and MR, we would have had the wrong interpretation of the data. But really, these two techniques are complementary. With the information of the oxygenation level, then we can properly infer about the mitochondrial function. Here again, there are a couple of pitfalls with the technique. First, you need to manipulate the carrier frequency or the excitation frequency. Again, you're 74, 75 ppm away from water. So you need to have a system that is going to allow you to change the carrier frequency. You also need a stimulus, either an ischemia or an exercise. Something that I did not mention is that there is a significant baseline distortion, which can complicate the quantification of the peak. And this is coming from the very large signal of the water. Finally, something that can be problematic with patient population is that you need to normalize the signal by using an ischemia for 6 to 8 minutes. We've used this technique in many patient population. That's usually not a problem, but again, there are some specific population where this might not be possible, and then your signal is really useless. You need these steps of standardization. Where do we see this technique going? Well, I think we have actually some hint, and there is a publication from Thomas Ju, so at UC Davis, that's actually from 20 years ago, where they actually tried to do this deoxymyoglobin mapping. So you have here a T1 weighted image of the calf, of the lower limb. And here, that's actually a matrix corresponding to the deoxymyoglobin signal. So they were trying to map the oxygenation level in the lower limb. This was done at 1.5 Tesla using a surface coil. So maybe, with improvement in the coil design and with a stronger field, this type of application would be very useful in PAD patients or type 2 diabetes patients. So there are some interesting applications with this technique. And there also, just as a reminder, even though the technique has been around for probably 30 years now, there are actually just a handful of labs that already use this technique. And yet, I think it provides some very important information. So there is still a lot to do with this technique. For the sake of time, I'm going to skip carbon 13, and I'm going to dive right into the imaging application in the skeletal muscle. And here, I'm actually going to start by talking about Rene Descartes. The reason being that when you're in high school in France, you need to study philosophy and you have to learn about his conception of the separation of the mind and the body. And I think if at the time my teacher had met an MR physicist, he would have tell him that this was just nonsense. What the MR field has been very good at is at bridging the gaps between the brain and the other organs of the body. So techniques that are developed in the kidney, the brain, or the skeletal muscle, they usually get translated very quickly to other organs in the body. And this is really one of the very nice aspects of the MR field, the sort of cross-pollination between, not between field, but between organs. And so here are a couple of applications which I think are of interest when we look at the skeletal muscle. So you can look at the muscle volume using T1 weighted images. So that's a fairly standard technique in the field. But you can also look at the structure of the skeletal muscle using diffusion weighted images, or DTI, for diffusion tensor imaging, which is illustrated here. And so you can actually really look into the orientation of the fiber in the skeletal muscle. And so actually Dr. Kent here has a project where they look at the effect of fat on the orientation of these fibers. And I believe also on the effect of aging on the fiber structure. Another technique that I think is of interest is this T2 mapping technique, which looks at the muscle activation. And so these data are actually from my doctoral work, where we looked at the T2 in the thigh before a knee extension exercise and right after. And so what you see after the knee extension exercise with the setup that we use is that you have a large increase in the T2 of the rectus femoris, suggesting that for the setup that we had, there was a very high activation of this muscle. But the scale is a little bit deceptive because we also had a large activation of the other muscles, but really the rectus femoris had a very, very large increase. And a couple of groups that tried to look into the origin of this signal, and they also reported this interesting relationship between T2 post-exercise and integrated EMG, suggesting that these T2 values, the change in T2 pre-post exercise, can be used as an index of muscle activation. The advantage compared to integrated EMG is that you can actually look deeper into the tissue. So you don't have to focus just on the superficial muscle. Now, the last application that I'm going to talk about is in terms of muscle perfusion and peripheral oxygenation. And in this slide, I'm sure all the physiologists in this Zoom call will look at this equation, and it's probably ringing a bell. That's the Fick principle, the Fick equation. And in this, so there are already probably 20, 30 publications looking at perfusion in the skeletal muscle using an MR. But I think this is a very elegant study. In this study, they actually very cleverly use different methods to be able to quantify all the parameters of this equation so that you could estimate muscle VO2. So they had 3 slices in the calf muscle. In the proximal slice, they measured blood flow through the popliteal artery using phase contrast MRI. In the central slice, they quantified tissue perfusion using pulsed ASL. And then in the more distal slices, they looked at the hemoglobin, the blood oxygenation using BOLD. And again, I thought it was a very clever design and indicating also, I think illustrating very well the sort of transfer that occurs between the different tissues. As long as the technique is developed, it can be used in any organs. BOLD is used for has been used for several decades to look at activation in the brain. Pulsed ASL has been used also in the brain and kidneys. Phase contrast MRI is widely used to look at for cardiac MRI. And I think it was a very clever application of all those techniques in the skeletal muscle. So the take-home message from my talk is really that MRI or MR spectroscopy MR in general is a very versatile technique that provides a lot of information that can be very insightful depending on your question. And you have again multiple techniques. All the ones that are checked here are actually either already in place at the hMRC or have been used by some of the researchers in the center. Probably the only component that would still require some development is really the measurement of tissue perfusion, which is actually quite a challenge in the skeletal muscle. And with that, I'd like to acknowledge the funding that my lab has received over the years from NHLBI and the family, and also would like to thank all the students that are in my lab and also the postdocs that are conducting research in my laboratory. The last year has been definitely challenging for a lot of us, and they've been doing an outstanding work trying to still get the project running. And so I wanted to highlight that in this talk. And I also want to thanks the kinesiology department and IALS for their support since I moved here. And with that, I will be happy to take any question.
Well, Gwenael, I can start you off with some pre-submitted questions if you'd like.
Yeah.
How do you ensure your spectrum is accurate in regards to removing compensating for noise since your subjects are moving during the exercise?
So it turns out that, okay, let me see if I can. Well, I guess I don't have to show the slide. First we had the coil that is attached to the calf muscle, which is our muscle of interest. So it turns out we don't get much, we don't lose signal, which would be one of the issue if someone is moving a lot in the scanner. Again, by wrapping up the coil around the calf, it turns out that we don't lose this signal. If we have a little bit of signal loss during the post-processing. We actually normalize the signal to the phosphate content because actually I'm going to show the slide. Let me share this one. If you have movement during the exercise, what happens is that you overall lose the signal. So all these peaks, I believe you can see the peaks. You can see the slide. So you would have a loss of signal from all these peaks will be much lower. And so we sort of standardize for that. And that would take into account the signal loss due to motion.
Are there any other questions for Gwenael or for Raja?
Now, what measures do we get out of the carbon, and how easy is it to switch between carbon and phosphorus?
So the volume was fairly low. What I heard is that how do you switch from carbon and phosphorus to look at both glycogen and phosphorus? I've never I've actually never done any experiments with glycogen. Um, and I think what you would have is actually you would need to repeat the exercise. Again, I skipped this part. One of the potential issue with measuring glycogen with carbon 13 is that it has actually a fairly low sensitivity. I was showing this table with phosphorus and proton. Carbon 13 is actually an exception where you have low abundance, low sensitivity. So what people tend to do is to inject some enriched carbon 13 through the vein. And even with that, you need very long acquisition time. So I think this is probably one of the, you would need to repeat the exercise. And also, I think you would only be able to measure these glycogen levels at low intensity exercise because you really need 10, 15, 20 minutes of acquisition before you have a decent spectra.
What is the time resolution of the phosphorus spectrum?
Oh, so the one I was showing is actually it's 5 minutes. Again, that's for one of our application. So you're talking about if I share my screen, this one. So yeah, this one, because we were trying to quantify this little alkaline pH, we have to obtain very good, the resolution of the spectra has to be very high. So this is 5 minutes, but for some of the spectra that I presented, like here, I believe this is every 6 seconds. So and you can still, you can see the main metabolites with 6 or 8 seconds in terms of the time resolution.
I have one more question. When do you use localized and non-localized MRS techniques?
So it depends on your question. If you want to look at flux, if I go back to, okay, let me show this. If you want to look at this ATP resynthesis through the main pathways, it's very difficult to use localized techniques. And I'm ticking a little. I'm a little bit. I don't like the term unlocalized technique. I know that Raja use it too, but so I will get back to that in a second. But so unlocalized techniques, that's when you want to study flux. Localized techniques, it's when you want to look at steady states. And the issue with localized techniques is that you need a longer, you don't have this nice time resolution. Like you cannot acquire one spectra every 8 seconds because you acquire a matrix. So usually you will need probably about a minute before you get a decent signal. So you lose the time resolution if you get the spatial resolution. And so if I have the time, I just want to show one slide about this term of unlocalized, which I don't like too much because usually people refer to unlocalized spectroscopy when you use surface coil. And so that's what I have here. Am I still sharing the screen?
Yes.
Okay. So they tend to refer to this surface coil where we have a high time resolution. And it's true that we don't know exactly where the signal is coming from. But what we do know is that this signal is coming from the tissues that are right underneath the coil. And so the reason I'm not a big fan of the term unlocalized is because this surface coil, when they were developed, so this is actually the original pair from Ackermann who developed this technique, these coils. That was designed to be able to actually get some localization of the signal. So the intent of the design of these coils was to actually localize the signal in a specific area of the body, because before that, that was just a whole organ or a whole body that was sort of investigated. So it is not localized in the way that more recently people have been thinking about, MR spectroscopy, but the title here is mapping of metabolites. So the intent is to localize the signal.
Great, thank you. Well, it's after 3, so I don't want to keep anybody any longer. So thank you so much, Gwenael, and thank you everyone for attending. Just letting everybody know our next seminar is scheduled for Tuesday, May 18th, where we'll be hearing from Genomics Resource Laboratory. And that will be the last seminar in our series, and then we'll be offering a few special seminars over the summer, so stay tuned. Goodbye, everybody.
All right, thank you.